What is spermatogenesis? Then what is spermatocytosis? What is the difference between spermiation and spermatid formation? The answer to all these questions is there in this video. Let's sort it out in 10 minutes. Spermatogenesis. Let's split the word. Spermato means spermatozoa and genesis means formation. So the process of formation of spermatozoa. Actual definition is maturation of male gametes in the wall of seminiferous tubules. Here, the male gametes are actually the spermatozoa. Primordial germ cells. These are the actual germ-line cells which are present in a fetus. These cells differentiate into spermatogonia in a male child and oogonia in a female child. After many stages of differentiation, the spermatogonia forms a mature sperm and the oogonia forms the mature ovum. Now let's take a male child is born. From birth to puberty, no change occurs. At puberty only, the process of spermatogenesis starts. So be very clear that from birth to puberty, there does not occur any differentiation in spermatogonia. What are the stages of spermatogenesis? There are totally 3 stages. The first is spermatocytosis, second is spermatid formation, and third is spermiogenesis. Spermatocytosis is actually the spermatogonium getting converted into a primary spermatozoa. In spermatid formation, the primary spermatozoa is converted to secondary and the secondary spermatozoa is converted into a spermatid. In spermiogenesis, the spermatid is actually getting converted into a spermatozoa. The time period is, for first stage it is 16 days, second it is 24 days and third it is 24 days. On a total it takes 64 days. Now let's take a primordial germ cell. The chromosome number of this primordial germ cell is 44XY. This primordial germ cells undergoes mitosis. So after mitosis, 2 cells are produced. In this video, we are taking 1 cell. So this PGC gives rise to spermatogonia by mitosis. The chromosome number remains constant, that is 44XY. So a child is born with 44XY spermatogonia. Then the male child attains puberty. Now what are the changes occurring to this spermatogonia? Let's see in detail. First it will be 44XY at puberty. Then this spermatogonia undergoes mitosis and then it forms primary spermatocyte. Mitosis, you have to remember, 2 cells are produced, but both daughter cells will have the same number of chromosomes. Here, dark type A spermatogonia are produced and light type B spermatogonia are produced. What is the difference between this dark and light type? Actually, they are microscopically different, but the chromosome number remains constant, that is 44XY. Now, this dark type A are actually the reserve cells for future division. This light type B only undergoes mitosis and helps in the formation of spermatozoa. Now, this light type B undergoes mitosis to form the first step, that is your primary spermatocyte. So, let's take only the light type B. This is having a chromosome number of 44XY. This is undergoing mitosis and hence it is forming primary spermatozoa. What would be the chromosome number? It is 44XY because in mitosis the chromosome number remains constant. That's why it is said that mitosis is actually not a reduction division. Now this primary spermatocyte undergoes meiosis 1. Remember that meiosis 1 is actually a reduction division. Reduction division means the chromosome number is halved. That is, totally the daughter cells will be having half the number of parent chromosomes. So the resultant secondary spermatocyte will have the chromosome 22X in one daughter cell and 22Y in the other daughter cell. So primary spermatocyte undergoes meiosis 1 to form secondary spermatocyte. This secondary spermatocyte then undergoes meiosis 2. This meiosis 2 is same as mitosis, that is, it is not a reduction division. So the parent number of chromosomes is as such retained in the daughter cells. As a result, one cell produces two cells. Here two secondary spermatocyte produces four spermatids. You can see the chromosome number is maintained. So finally we are getting 4 spermatids with 22X in 2 and 22Y in 2 spermatids. So this secondary spermatocyte is undergoing meiosis 2 to form a spermatid. This spermatid then undergoes maturation and then it forms the mature sperm. You have to note that the chromosome number in spermatid and the spermatozoa remains constant, that is 22 plus the sex chromosome. So finally we get 4 haploid spermatozoa.
Now let's see what is spermiogenesis. The spermatid getting converted to spermatozoa is actually known as spermiogenesis. This spermatid is actually a circular cell. So let's imagine a cell with its organelles. It has cell membrane, nucleus, Golgi apparatus, centrosome and mitochondria. How these cell organelles are getting modified to form a sperm is actually known as spermiogenesis, that is the spermatid into a spermatozoa. So first the cell membrane of the cell forms the cell membrane for the entire sperm. Now the nucleus of the cell forms the head. Then the Golgi apparatus forms the acrosomal cap. This cap covers 2/3 of the head of the sperm. The mitochondria of the cell forms the spiral segment of the middle piece. Then there is a proximal centriole and the distal centriole. The distal centriole forms the annulus and the proximal centriole forms the axial filament. Here light green is actually the annulus and dark green forms the axial filament. So head, neck, middle piece and tail are the four parts of a sperm. Now let's see the difference between spermatogenesis and spermiogenesis. Spermatogenesis is actually the complete process of formation of a mature spermatozoa from a spermatogonium. That is, it involves all steps, the mitosis, meiosis 1, meiosis 2, spermiogenesis, everything all together forms the spermatogenesis. Whereas spermiogenesis means transformation of the round spermatid into a spermatozoa. Now let's see the difference between spermiation and spermatid formation. Spermiation is actually the final step where the spermatids are released into the lumen of seminiferous tubules. The spermatid formation is a step where the secondary spermatocyte is undergoing meiosis 2 to form spermatid. What is maturation and capacitation of spermatozoa? The sperm is transported from testis to the epididymis. In the epididymis, it undergoes maturation. Full maturation is attained when it is mixed with the secretions of the prostate and seminal vesicle. Then what is capacitation? The final step of maturation of a sperm is actually known as capacitation. This process of capacitation takes place only when the sperm is ejaculated into the female genital tract. So, a sperm is fully matured only in the female genital tract.